Okay, let's get started. We're going to turn the, first off, thank you for joining us today for our ASO webinar sponsored by Phosphorex. On the screen, you see our whole team, very eager, get ready to start it for you. But let's go ahead and get started. Evan?
Good afternoon. My name is Evan Turek, the Business Development Director here at Phosphorex. I welcome and thank you for joining us today for our webinar titled Case Study, Development of Nanoparticles for the Systemic Delivery of Antisense Oligonucleotides to the Central Nervous System. The purpose of this webinar is to demonstrate how seraffinity polymeric nanoparticles were designed by Phosphorex in collaboration with CytoDyn to deliver ASOs to the central nervous system by targeting the transferrin receptor for receptor-mediated transcytosis. Before we start, a brief overview of our company. Phosphorex is a fee-for-service CDMO specializing in particle-based drug delivery solutions. Located in Hopkinton, Massachusetts, our state-of-the-art facility spans 30,000 square feet with an additional 12,000 square feet of cGMP space, which we expect to come online in 2024. Our team of technical experts have approximately 2 decades of experience offering comprehensive services from formulation feasibility and optimization to process development, scale-up, and tox batch. Phosphorex's core areas of expertise are lipid nanoparticles for the delivery of nucleic acids, polymeric nanoparticles for targeted delivery, and polymeric microspheres for sustained release. Our mission is to accelerate client programs and to deliver a tailored service experience as precise as the science. To introduce our speaker and my close colleague for today's discussion, Dr. Nick Boylan is the Director of Nucleic Acid Delivery here at Phosphorex in Hopkinton, Mass., where he leads a team of scientists and engineers that specialize in the development and optimization of novel drug delivery systems, including lipid nanoparticles and polymeric nanoparticles. Prior to joining Phosphorex, Nick directed the analytical and formulation development of a novel soft mist pulmonary inhaler while at Molecular Infusions, and contributed to the encapsulation of a wide variety of payload types with a proprietary nanomedicine platform called Accurins while at Bind Therapeutics. Nick holds a bachelor's degree in chemical and biomolecular engineering from Cornell University and a doctor of philosophy in chemical and biomolecular engineering from Johns Hopkins University. And now it is our great pleasure to introduce Dr.
Thank you, Evan. Good afternoon, everyone. Thank you for joining us today. It's my great pleasure to be presenting this work and providing some insight into the formulation expertise here at Phosphorex and some of this exciting work that we've performed in collaboration with CytoDyn. In terms of today's agenda, we'll be providing an introduction to antisense oligonucleotides as well as their delivery to the central nervous system. We'll also provide an overview of the seraffinity nanoparticle platform technology that has been developed in collaboration with CytoDyn, and happy to present our case study looking at the formulation design and optimization for the delivery of ASOs to the CNS via seraffinity. And as Evan pointed out earlier, Phosphorex really specializes in the preclinical lab services and in particular polymeric nanoparticles and microspheres as well as lipid nanoparticles. And moving forward, thanks to our partnership with Ampersand and investment that we received last August, we're building out our GMP facility as we speak, and this has really been a long-term goal for the company. So really excited to be offering those services in the future. And again, thank you to our CytoDyn collaborators for facilitating the data that we'll share here today. So as a quick intro to antisense oligonucleotides, so these are synthetic constructs, typically about 20 bases in length of single-strand DNA. So on the figure here, we have some of the common chemistries. On the far left, we have unmodified DNA RNA, and importantly, with the phosphodiester backbone. However, this chemistry is susceptible to nuclease degradation, and therefore it does not lend itself well as a therapy delivered kind of as a naked RNA and DNA. And so therefore, there's been a lot of work done over the years in exploring different chemistries to effectively stabilize and increase the potency of these molecules. So in the center of the figure here, we have some of the backbone modifications, and in particular, the phosphorothioate or PS modification, in which we've substituted a sulfur atom for the oxygen on the backbone. And this sufficiently stabilizes the molecule against nuclease degradation and can facilitate the half-life in vivo. Some other modifications include the morpholino and peptide nucleic acid technologies. And then finally, to further optimize sequence specificity and potency, there are various sugar ring modifications that can be performed on the 2' methyl location, and again, to enhance the potency of the overall molecule. So in terms of mechanism of action, here on the left-hand side, we have an illustration showing the typical process for expression of a genetic code from the nuclear DNA that is transcribed into a pre-RNA and subsequently processed into a messenger RNA. And upon export into the cytoplasm, this is effectively translated into the protein, the encoded protein. However, in a disease case where maybe there's been a mutation to that genetic code that leads to a dysfunctional protein, it may be desirable from a therapeutic perspective to block the translation of that protein. And this can be achieved at the RNA level through the use of antisense oligonucleotides, or ASOs. And so these ASOs can be designed to be complementary to various regions of the target RNA. And there are a few mechanisms of action. So upon binding the target RNA sequence, this RNA-DNA heteroduplex can trigger the recruitment of RNase H, which is an endonuclease that can facilitate degradation of the target RNA. Alternatively, just binding of the ASO to the target RNA can facilitate, through steric hindrance, the blockage of that protein translation, ultimately leading to down expression of that protein and correcting the underlying cause of disease. In terms of delivery to the central nervous system, there are a number of disorders that can benefit from ASO technologies, including spinal muscular atrophy, or SMA, ALS, Huntington's disease, and Alzheimer's disease. However, due to the presence of a blood-brain barrier, ASOs that are delivered systemically cannot freely penetrate into the CNS. And therefore, current therapies, as an example, Spinraza for SMA, require intrathecal administration or direct injection into the cerebral spinal fluid. But ultimately, less invasive routes of delivery are desired. Again, intravenous or sub-Q would be preferable, but require the development of effective drug delivery systems to overcome that blood-brain barrier. And therefore, nanoparticle delivery systems have been widely explored for ASO delivery to the CNS. And here we present kind of our case study for the utilization of seraphinity nanoparticles. So with regards to the blood-brain barrier, here we have a general schematic of some of the approaches that have been investigated historically. So on the top of the figure, we have a blood vessel and the lumen shown. And so at the top and bottom, we see the endothelial cells. And between those cells, we have the tight junctions that highly regulate the passage of soluble contents from the vessel lumen to the outer brain parenchyma. And some techniques that have been employed include the use of osmotic or chemical gradients to temporarily disrupt the tight junction. Alternatively, receptor-mediated transcytosis can be utilized, and that's the approach that we focused on for our studies, in which we target a receptor that's known to be present on the apical surface of the endothelial cells, such as the transferrin receptor. And upon binding, that receptor is internalized, and ultimately the nanoparticle can be released into the brain parenchyma. Certain nanoparticles may have the ability to penetrate independently of receptor-mediated endocytosis. Likewise, there's cell-mediated delivery, microbubble with ultrasound-assisted delivery. And then there have been some successful products that are simply implants, right, that can release the drug locally, let alone microneedles utilizing convection-enhanced delivery. So with regards to the Serafinity Nanoparticle Platform, this is a patented process in which during the formulation procedure, we can incorporate a wide variety of targeting ligands and non-covalently attach them to the particle surface. And for the purpose of CNS delivery, we decided to target the transferrin receptor, as it's been a widely investigated target, for the purpose of overcoming the blood-brain barrier via receptor-mediated transcytosis. And so in this case, we've been able to successfully incorporate transferrin at a wide range of amounts from 0 to 22% on a weight basis. And on the lower figure here kind of illustrates at a high level the formulation process. So nucleic acids, being very water soluble on their own, don't lend themselves well to encapsulation within polymeric nanoparticles. And therefore, we need to utilize a cationic counterion, either a lipid, polymer, or small molecule, to facilitate the encapsulation. And in this case, we're using PLGA as our degradable polymer. And again, transferrin is our kind of our ligand of interest that's going to facilitate that binding to the transferrin receptor. And ultimately, through the formulation process, which we'll describe shortly, we're able to generate a nanoparticle in which we have the ASO encapsulated. In this case, we're also using a fluorescent tag size 7 to facilitate in vivo imaging, and the transferrin is decorated on the surface of the particle. With regards to the Serafinity nanoparticles, here we've specified some target product profiles. With regards to the cargo, again, we have that size 7 conjugated ASO as a model payload, and due to the presence of that, the near-infrared dye, it'll be a convenient fluorescent tag for in vivo purposes. Particle size, we desire to be less than 200 nanometers. This is to facilitate long circulation time and penetration of the blood-brain barrier. For the particle distribution, we desire a PDI less than 0.3, ASO loading of greater than 1% on a weight basis, and likewise a range of transferrin contents, so sort of a low, medium, and high content. Here we have some additional details regarding that size 7 conjugated ASO. So first, we synthesized an ASO bearing a 5' amine group, and the sequence shown here is complementary to the long non-coding RNA MALAT1, which is widely expressed in a variety of cell types and tissues, so it's a kind of a convenient RNA target. And importantly, we have that 5' amine so that we can facilitate the conjugation chemistry, in this case utilizing size 7 dye that has a NHS ester, which is an amine reactive chemistry. Here we have some details regarding the size 7 labeling reaction. Starting with 85 milligrams of ASO prepared in a sodium bicarbonate buffer, we added a 1-to-1 ratio of the size 7 NHS ester and incubated at room temperature for 1 hour. We subsequently purified by 2 rounds of ethanol precipitation and lyophilized the final product. During the process, we monitored the UV absorbance at 260 and 750 nanometers in order to quantify the concentration of the ASO in size 7. And in the table, we have the calculated ratio. So we were able to confirm that in our reaction mixture, we had about a 1-to-1 ratio. And after performing the reaction and subsequent purification, we have 80% of our ASO conjugated with size 7. Regarding the formulation design and optimization, there were a number of critical formulation and process parameters that were considered. For the target ASO loading, we evaluated 7% on a weight basis, and this is relative to the mass of the polymer as well as the counter ion present. We also looked at transferrin contents in a low, medium, and high range. Since the ASO is relatively hydrophilic, it can be challenging to encapsulate on its own within a PLGA matrix, and therefore we utilized hydrophobic ion pairing, and in particular, small molecule ethyl laurel arginate, or ELA. So this molecule has the guanidinium group, which is a relatively strong base, and therefore at physiological pH is positively charged and can electrostatically interact with our ASO. And this is the mechanism that facilitates the encapsulation within our PLGA. Importantly, we fed the ELA to ASO at a specific ratio, and in particular, a nitrogen to phosphate ratio of 2. The polymer type in molecular weight is also important. So in this case, we're using Evonik's Resomere 502. So this is a relatively low molecular weight PLGA with a 50-50 ratio of lactide to glycolide. Whereas if we had used a higher molecular weight PLGA polymer, we would likely end up with larger particles that may be outside of our target range. Some other important considerations on the formulation include the solvent type. So we want good solubility of both the polymer, but also the counter ion, as well as the surfactant type and concentration, which is equally as important for generating kind of a stable, robust emulsion. In terms of the processing, your relative ratios of your aqueous and solvent phases are important during the emulsification process, as well as the homogenization technique. So in this work, we utilized probe sonication, which is very convenient for small scale test batches. However, Phosphorex has a wealth of expertise in alternative homogenization techniques, including microfluidics, as well as high pressure homogenization via microfluidizer. For the downstream processing, we relied on tangential flow filtration, or TFF. In particular, we use Replingen's KR2I system for processing these batches, which are relatively small scale. We also have Replingen's KMPI system for subsequent scale up of larger batches as well. Here we have our process flow diagram. So starting on the left-hand side, we have our solution prep of our aqueous solutions, as well as our organic phase. Next, we move into our double emulsion process using the probe sonication before evaporating the organic solvent, which effectively hardens the nanoparticles. And we can move into downstream processing utilizing that TFF in order to remove any unconjugated Cy7 ASO, and as well as any excess residual solvent. From there, we do a round of characterization before preparing our final sample. Here we have an overview of the double emulsion process. So first, we prepare our primary emulsion by pouring our Cy7 ASO containing inner water phase into our larger volume of organic phase, which includes our PLGA polymer. We utilize that probe sonication in order to homogenize that solution and prepare a continuous phase. So next we prepare our secondary emulsion by pouring that initial primary emulsion into a larger volume of aqueous. Importantly here, we have both surfactant as well as our transferrin targeting ligand. Again, we utilize another round of probe sonication in order to reduce the particle size associated with the oil droplets, followed by evaporation to solidify the nanoparticles. In order to perform the final purification, again, utilizing tangential flow filtration to remove any residual solvent and free unencapsulated Cy7 ASO, as well as to concentrate the final product. So in the illustration on the right, the feed reservoir effectively contains our nanoparticle suspension, and then utilizing the peristaltic pump, we circulate that suspension through our hollow fiber TFF column. The nanoparticles are circulated back to the feed reservoir. However, any lower molecular weight impurities, such as the solvent and an unencapsulated Cy7 ASO, can pass through the hollow fiber's permeable membrane and exit as the permeate or waste stream. Some important process parameters include the surface area of the column. So in general, largest surface area will allow a faster purification process. However, it comes at the compromise is larger surface area columns have a larger void volume, so you won't be able to concentrate your final product as high as you would using a smaller column. In this case, we're using a polyether sulfone column type, which has a 500 kilodalton molecular weight cutoff, and this is sufficient in order to allow passage of those lower molecular weight impurities while retaining our nanoparticles in the feed suspension. And lastly, we're utilizing DI water as a diafiltration buffer. So our final particles are suspended in DI water, which is convenient for subsequent analytical characterization. In our final dilution step, we will introduce 1x PBS for the final suspension. In terms of the analytical characterization, we monitor particle size at various stages of the process, and here we have the size for the final batches of particles that were prepared. Again, with the transferrin content ranging from 0, low, medium, and high. In this case, we had comparable sizes right around 120 nanometers with a size distribution or PDI of about 0.25. Ideally, in regards to particle size distribution, you're looking for PDIs of less than 0.2 or even better, less than 0.1. But with probe sonication, you tend to be kind of in this range, so somewhere between 0.2 and 0.3. For subsequent optimization down the road, we get much better size distribution using either microfluidics or high pressure homogenization. But again, for this proof of concept study, probe sonication is the most convenient tool. Looking at the surface charge or zeta potential, we observed an interesting trend in that the blank particles without any transferrin had a surface charge of minus 20 millivolts, and as the transferrin content increased, we had a more negative surface charge. And this is potentially due to the anionic nature of transferrin, which has an isoelectric point of 6, and therefore at neutral pH, it has an overall negative charge. We quantified the transferrin content using a modified BCA assay and observed a range from 0 to 22% on a weight basis. So fairly significant range there, and this was important for just establishing, what level of transferrin is required to potentially gain any benefits through the delivery to the CNS. And finally, we measured and quantified the size 7 ASO via UV vis, looking at 260 nanometer absorbance. And happy to report that our loading was fairly consistent between these batches, somewhere in the 4 to 6% range. And based on our input, this corresponds to about a 70% encapsulation efficiency. To assess the in vitro performance of these particles in the presence of cells, we went ahead and plated some neuro-2A cells, which is a marine neuroblast cell line, and treated with either free ASO, PLGA nanoparticles loaded with ASO but no transferrin on the surface, or the seraphinity nanoparticles, which contained ASO and had transferrin on the particle surface. There was initial incubation for 4 hours in Optimum at a target concentration of 3 micromoles with regards to the total ASO present. And then after 4 hours in incubation, we went ahead and added FBS and penstrep and incubated an additional 44 hours before isolating RNA, performing cDNA synthesis, and quantifying the mallet-1 RNA present by PCR. So as shown in the figure, with either the free ASO or the PLGA ASO nanoparticle lacking transferrin, there was about 60 to 70% presence of the mallet-1 RNA relative to an untreated control, or relative quantity. In comparison, the seraphinity nanoparticle, we observed 30% of the mallet-1 RNA present, or alternatively a 70% knockdown. So this was an interesting observation, indicating potential benefit of having the transferrin on the nanoparticle surface, which could potentially be impacting the the uptake into the cells and or potentially the intracellular trafficking of the ASO. With regards to our in vivo study design, we administered an IV dose of the 4 different nanoparticle types to CD1 female mice via the tail vein injection. And then 4 hours after injection, we performed the in vivo imaging for the Cy7 signal via IVUS. And upon completing the in vivo portion of the study, the mice were euthanized, carcass perfused, and the brain tissue collected for embedding in OCT and subsequent sectioning and staining before confocal imaging. And just a quick note, so the dose in this case was 6 mg per mil Cy7 ASO, which based on the roughly 4 to 6% loading is about 150 mg per kg particle dose. So here we have the results from the in vivo imaging study at that 4-hour time point. So the images here, they show the mice on the dorsal view, so we're viewing the backs of the mice, and the red signal corresponds to the Cy7 fluorescence. And so between all the groups, you tend to see, a good fluorescent signal kind of throughout the body, but there does tend to be a kind of a pronounced signal kind of in the head region. And when we draw a region of interest around the head and quantify the total radiant efficiency, we do observe an increasing amount of the Cy7 signal as the transferrin content increases in the formulation. Here we have the results of the confocal imaging. So for the mice that received the seraphinity nanoparticles with high transferrin content, we isolated the brain, embedded an OCT, froze, sectioned, and in this case, we stained with an antibody against nu-N, which is a neuronal cell marker. So in the images shown below, green represents the neuronal cells, blue represents cell nuclei via DAPI, and pink represents the seraphinity nanoparticles. So to facilitate the visualization, the yellow arrows indicate regions that have the Cy7, and in some instances, we do observe overlap with the green signal, suggesting potential co-localization of the seraphinity nanoparticles with the neuronal cells present in the brain tissue. So in summary, a proof of concept study was performed to investigate CNS delivery of ASOs via the systemic route. ASOs were efficiently encapsulated within PLGA nanoparticles via hydrophobic ion pairing. Cy7 was incorporated as the fluorescent label to facilitate in vivo imaging. The targeting ligand, transferrin, was successfully incorporated at low, medium, and high percentages on the nanoparticle surface. Based on in vivo IVUS imaging, higher fluorescence intensities were observed in mice, in the head region in particular, for formulations containing higher transferrin percentages. These preliminary results indicate potential co-localization of Cy7 ASO with neuronal cells, thereby supporting additional investigations. And overall, these results demonstrate that PLG nanoparticles can be leveraged to enhance the extrahepatic delivery of therapeutic oligonucleotides. Thank you for your attention. From here, I'd be happy to address any questions you might have. So if you haven't done so already, please enter those into the chat box. In addition, we have the email address both for myself and Evan. So, if any future questions or inquiries about Phosphorex capabilities, we look forward to speaking with you. Thanks again, everyone.
Thank you, Nick, for a great presentation. If Nick and Ben could come online so we could do the Q&A session, please.
Thanks, Kim. Hi, everyone.
So joining us today, we have our founder, president, and chief scientific officer, Dr. Bin Wu, who will drive our Q&A session with Nick. Dr. Wu?
Thank you, Kim. Thank you, Kim, for the introduction. Good afternoon, everyone. So let's get started with the Q&A session. So the first question I have here is, so transferrin binding peptide is competitive to transferrin or non-competitive. Any information on the KD, K on, K off for their binding to transferrin receptor?
Yeah, thanks, Ben. I can take that one. So yeah, definitely a great question. In our case, we were utilizing just kind of native transferrin and coating that on the surface. And certainly this would be competitive with any transferrin, right, that's just naturally present in the body. So this would be a competitive interaction with regards to the binding, right, of that transferrin receptor. So definitely some competition there. Yeah, we haven't studied yet to date the binding kinetics of the transferrin coated on the nanoparticle surface. However, yeah, we would anticipate a slightly different behavior. And just as a quick example, right, the nanoparticle itself has a much higher mass than a small, relatively small transferrin protein. So therefore, the on and off rates are likely going to be different. And this could be something that would be studied by a ViaCore or SPR. I do think that would be a very interesting study that could gain further insight into the behavior and potential further optimization of this nanoparticle system. But again, yeah, we haven't looked at that to date. So.
And the second question is, what is the expected stability of the end formulation product? What is the endotoxin levels? I can take this. So the nanoparticles, they are being made from a polymer PLGA. They are very stable. As long as you store them at like -20 degrees. And if you reconstitute them in a buffer, then obviously the PLGA will start to degrade, and the product, the ASO will start to release. So, and then the colloidal, in terms of colloidal stability, these nanoparticles are well formulated, and then they have good colloidal stability, and they don't tend to aggregate after the reconstituted. In terms of the endotoxin level, we haven't not measured it specifically, but we have no issue with the animal study.
Yeah, maybe I'll add Ben real quick, right, with endotoxin. Yeah, we do have a lot of experience with other programs in evaluating endotoxin burden. And I think just at a high level, what we tend to find with regards to contamination, it's typically not process related, but associated with raw materials. And so therefore, whenever possible, right, we like to use the highest grade raw materials as possible, right, whether they're kind of USP and certified to be a low endotoxin, for example. So I think that's one critical step. And then when we do ultimately run our fabrication process, there are certain precautions that we can put in place to minimize any contamination of the formulation. But yeah, certainly an important consideration.
Thank you, Nick. So the next question is, what other organs did the cargo distribute to, in addition to brain? Nick, you want to take that?
Yeah, absolutely. So I think nanoparticles in general, right, inherently distribute to other organs, and in particular the liver, as well as some uptake in the kidneys. So in this case, yeah, we have typically about 60% uptake in the liver, and then maybe about let's see, about 25% in kidneys, and then the rest of the dose is kind of spread across the remaining organs, kind of being the lungs, the heart, the spleen, for example, as well as the brain tissue. So yeah, that's kind of the general distribution. I think in this particular application, right, we utilize transferrin, but with our formulation technology, we are amenable to utilizing other ligands for potentially targeting, whether it's CNS or other tissue types. And for example, Cytodyne has explored delivery, for example, to the spleen and also immune cells in particular. So definitely an area of ongoing kind of research for us.
Yeah, if I could add, so unlike lipid nanoparticles, where the usually the liver is the main target, is the main destination, right? The probably 90 or 95% of IV injected, the NMPs would go to liver. But here we showed that only 50-60% of the nanoparticles go to liver, so which allowed some of the to some degree extrahepatic delivery of the oligonucleotide. And the next question is, so what advantages might degradable polymeric nanoparticles have over lipid nanoparticles in terms of penetrating the blood-brain barrier? Maybe I can start first. So polymer nanoparticles are more stable than lipid nanoparticles. So therefore, they can protect the therapeutic payloads like mRNA or siRNA more effectively. Another advantage is it is easier to change the surface characteristics of polymer nanoparticles to vary the biodistribution and also to enhance the accumulation of the nanoparticles in the brain tissue. For example, we in this case we showed that we can attach transferrin on the surface of the nanoparticles, polymer nanoparticles, without the need of a chemical conjugation, right? You wouldn't be able to do this with lipid nanoparticles. And another advantage is the polymer nanoparticles can provide some of the sustained release, long-acting effect. So that could effectively reduce the dosing frequency, so that could help enhance the patient compliance. Yeah, I don't know if you have anything to add, Nick, to this question.
Yeah, I think that's a good summary.
Let's see. So the next question. Yeah, go ahead.
So regarding the sonication process and kind of the energy, right, put into that emulsion, and would there be any negative effects on the drug stability? Yeah, so it's definitely a good question. And in our case, we certainly perform that step on an ice bath, right? So trying to keep the temperature from increasing, because during that process, right, without any temperature control, the formulation temperature can increase over time. And certainly if you apply too much energy, there could be degradation to not only the cargo, but also the polymer. So in our case, like kind of trying to balance the amount of energy applied. So this is typically in short bursts. So, maybe 5 seconds on of sonication power, and then with maybe 30 seconds off. And again, being an ice bath, using enough sonication power and time to reduce the oil droplet size into our target range, and kind of not doing unnecessary additional time on the sonication, right? So that's how we can kind of balance that energy input. And in our case, with regards to whether or not the ASO was still intact, we did confirm in vitro that we observed a knockdown, right, of the target MALAT1 RNA using the affinity nanoparticle. So I think at least that's one piece of information indicating that we do have intact ASO in that final product. But certainly with a given project, right, we could do a more thorough analytical investigation to confirm, just one example by HPLC and even better by mass spec, what is the, is that ASO intact? And for example, is there any kind of degradation to that molecule? But in this case, yeah, these chemistries are relatively stable. And so, yeah, we're fairly confident that at least a good portion of this ASO is remaining intact during this process. And maybe the one other kind of the follow on with that would be, we also have access to more gentler mixing technologies. In this case, I think microfluidics, right, would be preferred. And it really just using those microfluidic chips and in likely different solvent types, we could achieve similar nanoparticles, but under a much gentler formulation condition, for example. So yeah, great question. Appreciate that one.
Mm-hmm. And the next question is, from the formulation procedure scheme, I did not see when you add the ELA molecule, if I remember it right, for the nucleic acid encapsulation.
Okay, yeah, I can take that one as well. So in this case, and just to kind of go through it really quickly, so we utilize double emulsion. So first we solubilized our ASO at a relatively high concentration in what we call the inner water phase. And this is so that we don't have to use much volume to achieve our 7% loading on a weight basis. So again, we have that ASO dissolved at high concentration, and then we dissolve both the PLGA polymer and ELA into our organic solvent system. And so when we run the first sonication step, we're introducing the ASO, so a relatively small volume, maybe 100 or 200 microliters into roughly 1 or 2 mils of this organic phase. And that's sonicated before going into our second emulsion step, which involves a much larger kind of outer aqueous phase.
And the next question is, what is the blood T-half of the particle in the mouse?
Yeah, from my end, I guess we haven't done like a long-term, like kind of PK analysis, right, of the half-life, the serum half-life. I think in general, these nanoparticles, depending on their surface coating, can be designed to circulate for a long time. But yeah, I think for us, that would definitely be a follow-on study.
Another question is, can you incorporate the ASO into NMPs and have them cross the blood-brain barrier?
Yeah, thanks, Ben. I can take that one. So I think certainly, yeah, lipid nanoparticles, LNPs, are the leading non-viral gene delivery system. Historically, right, they've been used primarily for maybe siRNA, messenger RNA, plasmid DNA, basically therapeutic cargoes that are susceptible to nucleus degradation, right, and aren't necessarily taken up into cells just by the naked molecule. So therefore, with ASOs in particular, they haven't necessarily relied on like a lipid nanoparticle for delivery due to their stability and ability to enter cells on their own. However, just given the nature of an ASO and that phosphorothioate backbone, for example, it still has that high negative charge density. And so using the ionizable lipids that have been developed for siRNA and messenger RNA, you can definitely encapsulate ASOs within a lipid nanoparticle system. And in Phosphorex, we do a lot of work in the lipid nanoparticle space, primarily for encapsulation of messenger RNA and plasmid DNA. So these technologies can be merged, and we also have a lot of expertise in conjugating targeting ligands to the surface of lipid nanoparticle as well. But kind of to date, yeah, we haven't necessarily explored lipid nanoparticles for delivery systemically to the CNS. So yeah, that's something we haven't looked at, but I think certainly would be an exciting area to look into further.
Mm-hmm. Another question is, I don't really understand how you incorporate transferrin on the surface. Can you go over a little bit more on you add transferrin in the second emulsion step. Am I correct? I can take this. So at Phosphorex, we have developed a process which allows us to incorporate the ligand, polymer ligands and or protein ligands on the surface of polymer nanoparticles without having to do chemical conjugation. And so the way we did it is we dissolved that, say, in this case, the transferrin in the solution with some other surfactants like transferrin, like polyvinyl alcohol. And then doing this, then we run the emulsification, either single emulsion or double emulsion process. And then we followed by the solvent evaporation and washing step. And we discovered that, with this specific formulation and process, and these certain ligands can be trapped on the surface, through interpenetrating network. So no matter how you wash them, how many times you wash the particles, they would stay on the surface. So it's partially kind of entangled into the molecular structure, the particle structure. But with the ligand, most part of the ligand, sticking out on the surface. So that's the process we call interpenetrating network. So using that process, we were able to incorporate several targeting ligands on the surface of polymer nanoparticles. Yeah, we can discuss it more in details if you wanted to email us, then we can discuss further.
We also have their contact information, so we definitely could reach out, have a separate conversation. I think that's all the time we have today for questions. Thank you both very much. There are some really great questions that came in, but obviously we can't get to all of them today, but we can get to them via email, should that be the case. But again, thank you for joining us today for our webinar. A recording will be emailed to you within 24 hours. Have a great rest of the day, and we look forward to seeing you in September for our next webinar. Have a good day. Thank you.
Bye.
Thanks, everyone.